So at this point in the class, what we want to start doing is thinking about how we can apply the concepts we learned about amino acids and peptides to considering how proteins are constructed. So just as a reminder here, when we're thinking about amino acids and peptides, right? So we're talking about single amino acids, which we know have an amine terminus. We go through an alpha carbon, which has an R group attached to it, and then we have a carboxyl terminus, so a C terminus. And when we have multiple amino acids that combine together through a condensation reaction, we end up forming a polypeptide chain, which we still have single amino acids, but now instead of attached to an oxygen, we're attaching to another nitrogen through this peptide bond. We have now a second R group, another carbonyl. We attach to a nitrogen through another peptide bond, our third R group, and we proceed in this manner, right? So when peptides form, we form these peptide bonds, and that gives these new molecules, these new polypeptides, the properties of each of the individual amino acid side chains. So when we talk about protein structure, we break it up into three categories. We have primary structure, we have secondary structure, and we have tertiary structure. And we abbreviate these 1, 2 degree, and 3 degree. Okay, so our primary structure consists of the amino acid sequence of a protein. Amino acid sequence. Okay, so when we think about the properties of the primary structure of a protein, what we're really thinking about is the functionality that it has because of the sequence of amino acids. So for example, if we have a peptide that's alanine, valine, lysine, right? So the primary structure has everything to do with the side chains. So here we have a CH3. Valine, we know we have a CH and these two CH3s hanging off. The lysine, we have CH2, 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 and then we have this NH3 hanging off down here. Right? So when we talk about primary structure, we are talking about the amino acid sequence and the chemical and physical properties that are conferred to this polypeptide chain because of the side chains. Okay? When we're thinking about secondary structure, I'll change colors here. When we're thinking about secondary structure now, what we're thinking about is how the backbone of a polypeptide chain can adopt certain structures. Okay, and the two really big structures that we're going to care about are beta sheets and alpha helices. Okay, so these two secondary structural elements or secondary structural motifs are going to have dedicated videos to discuss them. But so just in general, when we're thinking about secondary structure, what we're really thinking about are these conformations that fold, these structures that fold within the polypeptide chain. Okay, so for our beta sheets, when we look at a cartoon diagram, we see them as these two-dimensional arrows. Okay, and they're oriented such that the arrow points from the N terminus to the C terminus. Okay, so when we think about our secondary structure, if this line represents the polypeptide chain from the N terminus to the C terminus, then whenever we see this arrow, that means that all of the amino acids within this region of the polypeptide chain are part of a beta sheet. Okay, our alpha helices we represent as cylinders. Okay, and again, these are always going to be oriented from the N terminus to the C terminus. Okay, so within the polypeptide chain, if we have a cylinder, this means that these amino acids, this region of amino acids, are part of an alpha helix. Okay, so if we continue on here, right? So these amino acids would be part of a beta sheet. These amino acids would be part of a beta sheet. These amino acids will be part of an alpha helix, right? So this is a cylinder. Okay, and then we finish up over here at our C terminus. So we can show these diagrams of the polypeptide backbone with these cartoons, or these images drawn in that represent secondary structural elements. And we always show alpha helices as cylinders and beta sheets as arrows. Okay, so tertiary structure then, we'll start it off down here again. So tertiary structure is thinking about the interactions between the secondary structural elements. So it's how the secondary structure compacts itself to form the true three-dimensional structure of the protein. All right, and this drawing that I'm going to give here, that I'm going to show you here, probably doesn't do justice to what's really happening. But what I'm going to try to do is kind of show how these secondary structural elements might combine. Okay, so I'm going to go ahead and label this as beta sheet 1, beta sheet 2, and beta sheet 3. Right, so the idea here is that these three beta sheets can combine into one full sheet. So, right, if here we show the first one, okay, from here we kind of go on and we have an alpha helix. So I'm going to make a curve here. We then have an alpha helix, okay, and then we then transition into the second beta sheet, and then we transition into the third, and then finally we would go on to our last helix here, which maybe ends up pointing this way. Okay, and then off to our C terminus. So the idea here is when we're thinking about tertiary structure, what we're thinking about is how these individual secondary structural elements interact with each other. And as we're going to talk about in, I guess, towards the end of this unit, is that the interactions between secondary structural elements really boil down to chemistry between the side chains. So tertiary structure boils down to side chain interactions, where secondary structure up here really boils down to interactions between the backbone of within the backbone of the polypeptide chain. Okay, so the next few lectures are going to focus a lot more on secondary structure, and then we'll kind of finish up with thinking about how the beta sheets and the alpha helices can interact in a way to form a compact protein structure that can have a very specific function.